Hello, and welcome to the Drug Discovery World podcast. My name is Giles, and I'm here to take you through another topical and insightful article from Drug Discovery World. Today's episode is taken from an article in our winter 2016-17 issue, and is part one of two episodes, titled Translational Chemical Biology, Gap Assessment for Advancing Drug Discovery, Development, and Precision Medicine. The article was written by Dr. Mukund Chogard, Dr. Michael Liebman, Dr. Gerald Lushington, Dr. Stephen Naylor, and Dr. Rathnam Chagaturu. With so many contributors to this article, we'll be talking through their backgrounds at the end of part 2, the second episode. So now onto the main article. Translational Chemical Biology, Gap Assessment for Advancing Drug Discovery, Development, and Precision Medicine. Part 1. As yesterday's lead molecule enters today's clinical trial, the standard operating script seems to call for product developers and clinicians to push away the originating basic scientists, lest their lofty and practical ideals disrupt a delicate balance of strategic compromise. Indeed, many scientists may not understand the competing push and pull of efficacy versus toxicity and quality versus production costs. They do, however, know the molecule in question intimately. Their insight into subtle vagaries of the incumbent chemical biology can prove invaluable in clearing unexpected hurdles. The sophisticated techniques they applied to advance the molecule this far have not lost their analytical magic. They may very well save your bacon. Translational research should never be considered a relay race handoff, but rather a march together, hand in hand, towards a shared victory. In order to foster pragmatic collaboration, we endeavour to examine key technologies that not only enable early-stage discoveries, but can also bridge the late-stage pitfalls that may threaten promising drug candidates on the path to market. Translational chemical biology sits at the nexus of chemistry and biology. While the application of chemical biology principles helps in the designing of a bona fide chemical probe, it is translational chemical biology that helps translate basic research into meaningful clinical applications. This trajectory of chemical biology to its applied domain is interdisciplinary, and one that is yet to be mastered by academia. Successful outcome of any translational chemical biology effort is guided by several key factors: the impact of precision medicine, the reality of the valley of death, and what natural compounds and their clinical use can teach us. These issues are critical to improving drug development and lowering barriers to their translation into clinical utility and commercial value. Target-based drug discovery, a solely bottom-up rather than top-down approach, limits effective translation, particularly when viewed as a progression from laboratory to clinic. Observational therapeutics, guided by the principles of reverse pharmacology, the bedrock of traditional medicine but lately forgotten by the pharmaceutical industry, holds the key to the bench-to-bedside holistic seesaw pendulum. Current drug approvals. While biotechnological advances, genomics, and high-throughput screenings or combinatorial and asymmetric synthesis have long promised new vistas in drug discovery, the pharmaceutical industry is facing a serious innovation deficit. The costs of drug development have escalated, the number of drug withdrawals has increased to historic highs, and the transition from bench to bedside has been long and arduous. It is instructive to evaluate the valley of death conundrum from the perspective of the drug approval process and drug product output. In 2016, the US FDA approved 22 new molecular entity (NME) and new biologic license applications (BLA) from a total of 41 actual filings. This was the worst year since 2010, when only 21 NME and BLA drugs were approved from 23 filings. New drugs approved in 2016 included 8 fast-track designations, drugs that address unmet medical need. 7 were deemed breakthrough treatments, clinical evidence indicated substantial improvement over other therapies. 15 received a priority review, 6 months instead of the standard 10 months, and 6 were accorded accelerated approval for drugs that demonstrated superior performance over existing therapies for life-threatening disease indications. The FDA has introduced these designations over the recent past in order to expedite the drug discovery and development process and hasten superior drug performance candidates to market in order to meet the urgent, unmet medical need. Janet Woodcock, director of the Center for Drug Evaluation and Research at the FDA, noted that a lower than average number of novel drugs were approved in 2016 by the FDA. However, she claimed that the quality, impact, and unique contributions to improved patient medical care was extremely high for these approved NME and BLA therapeutics. For example, 8 of the 22 approved drugs were first in class, representing new drug target mechanisms of action. In addition, 9 approved drugs received rare or orphan disease status. This designation applies to disease indications that afflict less than 200,000 individual patients, and is significant because oftentimes such patients have no available therapeutic options. Woodcock argued that more important than the quantity of novel drugs approved in 2016 is their medical value and the important new roles they are serving to advance patient care. The quality versus quantity argument has some validity given recent trends in marketing new drugs that possess modest, incremental performance capabilities compared to other same-in-class drugs, for example statins. However, given the vast number of disease indications with minimal or ineffective treatment options, it is imperative to increase the quantity as well as the quality and effectiveness of new drugs coming to market. In that regard, it is revealing to review the past 70 years of new drug activity at the FDA. There has been a slow but notable trend in the increased number of new drugs introduced into the US market by the FDA on a per-decade basis. For example, in the 1940s, 192 new drugs were introduced. Subsequently, 1950s, 200, 1960s, 151, 1970s, 170, 1980s, 217, the 90s, 311, the 2000s, 235, and projected for 2010 to 2019, 321. Based on these approved drug numbers and trends, proponents of the pharmaceutical sector argue that the drug discovery and development process is functional and innovation and creativity is alive and well. There is also an argument made that the valley of death is overhyped and overemphasized. Translational Chemical Biology, the knowledge deficit. Since the launching of the National Institutes of Health, NIH, Roadmap Initiative, NIH continues to invest millions of dollars to harness the repertoire of druggable therapeutic targets that have come to life from the Human Genome Sequencing Project. This effort made the academic scientist appreciate, if nothing else, the ingenuity of its brethren from the pharmaceutical industry in the discovery and development of drugs. NIH's investment resulted in the development of exciting assay technologies, brought high-throughput screening to the academic corridors, thousands of compounds screened, millions of data points generated, data analytics became the mainstream language, made inroads in optimizing screen hits, and strengthened chemical biology as a discipline in designing chemical probes. While this list of accomplishments comes across as seemingly impressive, the chemical probes discovered through this effort almost never saw the light of day, especially from a clinical perspective. Inability to secure intellectual property rights and the dearth of medicinal chemistry expertise proved too steep a hill for the academic scientist to climb. Lack of translational medicine experience contributed to a non-starter in what makes a therapeutic target hit leap from being an in vitro active compound to become a candidate with clinical potential. This is translational chemical biology, and is still in its infancy in both academia as well as in many budding biotechnology companies. NIH has made a strategic effort in addressing this knowledge deficit by instituting Small Business Innovation Research Grants program to bring pharmaceutical experience to entrepreneurs and support research and development that has a strong potential for commercialization. Even with research dollars available, it is incumbent upon the chemical biologist to learn and implement the principles of translational medicine as discussed in the rest of this episode. Deploy modern tools to augment scientists' repertoire. The mismatch between academic efforts and that of industry is clearly of some concern. The pharmaceutical sector may argue that the drug discovery and development process is already efficiently aligned. We would suggest that the efforts of the academic community may need to be somewhat realigned if they are to participate in contributing and improving the drug discovery and development process. In part, this can be done by a more careful consideration of implementing a useful translational chemical systems biology approach. Modern-day combinatorial chemistry, CombiChem, high-throughput synthesis and screening, were amenable only to production of libraries of flat, planar molecules with remarkable lack of chirality and scaffold diversity. Creative biology has been retrofitted to mundane, uninspired and uninspiring chemistry. Moreover, plant-derived molecules with embedded chirality or synthesized with high stereospecificity have been neglected over many years due to proclaimed difficulty in securing intellectual property and perceived problems in obtaining large quantities of materials. Critics suggest we have become high throughput in technology, yet have remained low throughput in thinking. Post-marketing failures of blockbuster drugs have become major concerns of industry, leading to a significant shift in favour of single to multi-targeted drugs and affording greater respect to traditional knowledge. The typical reductionist approach of modern science is being revisited over the background of systems biology and holistic approaches of traditional practices. Translational chemical biology for drug discovery is useful. It is extremely imperative that the newer tools and techniques are brought to the forefront. Sophisticated organic synthesis must still be the order of the day. Professor Robert Burns famously stated, "The art and science of chemistry is one that is more easily exemplified and epitomized than it is articulated and summarized." We discuss herein some approaches and technologies that can indeed pay rich dividends in discovering and progressing a bioactive from mind to marketplace. Reverse pharmacology observational therapeutics. Reverse pharmacology is the development of drug candidates by validation of clinically documented experimental data on plant-derived natural products. In this approach, scientists and clinicians integrate bedside documented experiential clinical data from plants used in Indian traditional medicine and from Chinese traditional medicine. Reverse pharmacology involves isolation, structure elucidation, and biological mechanism of action studies on active compounds and assessment of novelty. One can expand on the already known activities of natural products derived from reverse pharmacology by selective functionalization, generation and derivatization of privileged structures, and conducting selective studies on preclinical development. As the starting point are compounds with established safety and efficacy, one can then establish early proof of concept in humans and differentiation. Scientifically validated and technologically standardized botanical products may be explored on a fast track using innovative approaches such as reverse pharmacology and systems biology, which are based on traditional medicine knowledge. Many modern drugs have their origin in ethnopharmacology and traditional medicine. Indian Ayurvedic and traditional Chinese systems are living great traditions. Ayurvedic knowledge and experiential databases can provide new functional leads to reduce time, money, and toxicity, three main hurdles in the drug development. Extensive information on Ayurvedic medicine research, clinical experiences, observations, or available data on actual use in patients can be used as a starting point. Principles of systems biology, where holistic yet rational analysis is done to address multiple therapeutic requirements, are coalesced with chemistry. Since safety of the materials is already established from traditional use track records, it is necessary to undertake pharmaceutical development, safety validation, and pharmacodynamic studies in parallel to controlled clinical studies. Thus, drug discovery based on Ayurveda follows a reverse pharmacology path from clinics to laboratories. Renaissance of plant-based natural products. Natural products have been historically regarded as rich sources of novel molecules of broad utility and architectural complexity. The natural world is a source for inspiration for chemists and biologists. The exquisite and varied architecture of natural products provides a rich palette for discovery. Natural products can be considered pre-validated by nature, having been optimized for interaction with biological macromolecules through evolutionary selection processes. Embedded in these bioactive natural products are a number of diverse chiral functional groups that are potential sites for protein binding. This diverse source of novel active agents serves as leads or scaffolds for elaboration into desperately needed efficacious drugs for a multitude of disease indications. The chemical entities present in natural product may or may not be useful as a drug, just like the hits from target-based high-throughput screen. However, their activity can be enhanced either by making modifications or combining with other similar pharmacological activities that will make these hybrid molecules more drug-like. Scientists must aim to reconfigure products into chemical, hybrid, molecular Legos, and to screen the deck of diverse compounds against targets. Investigations should centre on, first, expansion of the already known activities of natural products derived from reverse pharmacology by selective functionalization, generation, and derivatization of privileged structures and conducting selective studies on preclinical development. Second, designed organic synthesis of high recognition libraries focused on specific biological targets. And third, synthesis of building blocks, scaffolds, or high-value intermediates. Expanding chemical space. The ability to easily access new chemical space, however defined, is a major challenge for discovery chemists. Although advances in diversity-oriented synthesis have made great strides towards expanding the accessibility of synthetic compounds that have high levels of diversity, there is room for further improvement. The diversity expansion inherent in transformations that mimic the metabolism of small molecules and natural products can, in conjunction with modern synthetic organic chemistry, provide a new direction in the pursuit of natural product-derived substances. Simple and well-controlled transformations, oxidation, halogenation, and alkylation, can afford compounds that have unique structures that possess a wide range of physiochemical and biological properties. Indeed, such biomimetic transformations are the source of remarkable levels of diversity in natural products and investigational agents. The enthusiasm for natural products discovery has sometimes been dampened by difficult synthesis. A significant disadvantage of natural products, with the exception of those derived from fermentation, is the draconian organic synthesis medicinal chemistry effort required for commercialization or future functionalization. In many cases, the natural product compound has not been available in sufficient quantities for various biological assays, thereby limiting their exploration. Plant-based natural products are at times available in greater quantity. Hybrid molecules. Recently, the concept of hybrid molecules has gained currency. Couplings of diverse molecules such as artemisinin and chloroquine with vitamin K3 have generated new molecules with effectiveness in oncology. The therapeutic properties of several natural and synthetic products can be modulated by such hybrids, possible to get alternate therapeutic indications, and hybrids can be structural or functional. Flow chemistry. In recent years, flow chemistry has become a powerful tool for the rapid optimization and refinement of organic reactions and processes. Most are typically employed on a microscale for proof-of-principle studies. Modifications allow flow to be also used for bulk production of candidate compounds of interest. The three most common variants of flow chemistry are microflow, mesoflow, and tethered reagent flow. Flow offers many advantages over conventional batch chemistry. Pressurization allows reactions to be formed at temperatures exceeding the boiling points of solvents. Multi-step reactions can be performed in continuous sequence, and gaseous reagents can be incorporated into reaction sequences with limited effort. The processes are also amenable to rapid automation, improving throughput in the discovery phase. Additionally, improved impurity profiles, one of the benefits of the controlled mixing heat transfer characteristics that the method offers, is typically observed, as in the case of microwave-mediated synthesis. The flow approach allows rapid assembly of diverse libraries of building blocks for synthesis. One can envision its use in optimizing a number of intra- and intermolecular transformations. Bound reagents. The use of bound reagents and scavengers is an emerging technology frequently employed in streamlining reaction setup, workup, and purification. A key advantage of the use of bound reagents is the ease of conversion of batch reactions into flow format. There is a huge variety of commercially available and made-for-purpose resins that can be employed. These include oxidants, reductants, bases, acids, and bound or encapsulated catalysts. Chemoinformatics. It is tempting to regard molecular modelling and chemical informatics as predominantly basic science tools, abstract engines for hypothesis generation, rather than rigorous techniques for addressing the advanced, fine-tuning requirements of translational drug optimization, wherein promising leads are tweaked for efficacy, selectivity, toxicity, and deliverability. In truth, however, this preconceived notion of computational analysis has little pragmatic or logical basis. When both the methodological strengths of computational chemistry and the specific requirements of translational science are considered, it is actually easier to argue that modelling and informatics have significantly greater aptitude for the later stages of therapeutic optimization process. Indeed, rigorous computational exploitation of data on hand may provide a critical helping hand for navigating the valley of death. To understand the strengths of computational chemistry at various stages in the drug development process, let us consider how such tools are used. Early in the basic science stage of a chemical biology investigation, modelling and informatics methods may provide glimpses of factors that could play roles in a given physiological process and the modulation thereof. Such early phase investigations, however, tend to be idea-rich yet data-poor. Without a solid basis of reliable analytical measurements or empirical understanding, such speculative glimpses bear little predictive value. The veracity of computer-generated observations may be difficult to assess in such an environment, or, if the analysis elucidates real phenomena, it may not be immediately clear that these features have a strong bearing on the key issues of medical interest. By contrast, in the later translational stages of an investigation, one is almost always better armed with extensive data and empirical evidence. Much of this insight can be directly leveraged to intelligently select computational protocols that can be shown to reproduce what is already known, and to foster effective method and parameter collaboration that optimize the methodological fidelity for de novo calculations. As a practical example, the early stage application of molecular modeling and chemical informatics analysis towards hit elaboration will typically be constructed by either the small size or the great heterogeneity of bioactivity data. Later on, after many laboratory studies have compiled homogeneous datasets spanning a good number of close structural analogues to the lead compound, this basis of knowledge enables far more rigorous computational perception of subtle SAR trends. This perception can foster accurate refinements in deliverability, efficacy, target specificity, toxicity, or a host of other important concerns. When the existing data contains biomolecular structural insight, for example receptor crystal structures or NMR, one can employ data trends to train detailed comparative binding energy, combine models that prioritize specific ligand-receptor interactions according to favorable or problematic contributions to the observed SAR. If the data do not contain receptor structure information, computational methods can still prove very helpful. Machine learning methods are exceptionally well suited for parsing the properties of specific hits or lead analogues and contrasting those features against therapeutic efficacy, selectivity, or toxicity assessments. Such analyses can pinpoint the precise combination of desirable molecule attributes that favour some candidate ligands over others, and can suggest opportunities for further refinement, as well as plausible trade-offs, such as those between efficacy and drug deliverability, or between pathogen cytotoxicity versus host toxicity. An impressive array of lead optimization issues may be plausibly addressed through protocols that rely at least in part on molecular modelling or chemical informatics. Such issues include, among other things, many aspects of oral availability, volume of distribution, blood-brain barrier permeability, pharmacodynamics, and pharmacokinetics. Computational techniques are also relied upon for the design of non-drug delivery formulation components suitable for stabilizing a therapeutic or facilitating its integration into pill form. Great reliance may also be placed on modelers and informaticians in Phase 2 and Phase 3 clinical filings for the assessment of trace component toxicity. Specifically, while there is no substitute for careful and incontrovertible in vivo toxicology testing of the primary therapeutic ingredient or any drug formulation, late-stage scrutiny of medical formulations places significant emphasis on prospective complications that may arise from trace contaminants present in the formulation as synthetic impurities, or that may emerge due to metabolic conversions. Since trace impurities and metabolites may number in the dozens or hundreds of distinguishable chemical entities, few regulatory boards require that they all be subject to exhaustive toxicology testing. However, some quantifiable risk assessment is nonetheless required, and a commonly sought level of diligence entails computational assessment of any toxic or mutagenic indications, or any potential adverse consequences. Requested evaluations rarely require unanimous declaration of non-toxicity across a broad range of predictive measures. Instead, approval is generally granted if the petitioner can provide assurances from at least two unrelated computational techniques that the threshold of adverse effect for any given trace entity is below the quantity presented in a standard dose, or in the metabolic process thereof. A significant number of free internet-accessible computational resources are available for such determinations, including packages based on either QSAR model training or on empirical toxic effect rules. One final area where chemical informatics is blending with sophisticated new bioinformatics methods toward enhancing translational aspects of pharmaceutical practice is in precision medicine, whereby new software tools are emerging to exploit personalized genome profiling in combination with extensive chemical biological data in order to predict toxicity and efficacy on a patient level. And that's the end of part 1 of Translational Chemical Biology, Gap Assessments for Advancing Drug Discovery, Development and Precision Medicine. In part 2, we'll be discussing process chemistry, route selection, drug metabolism, the case of the cytochrome, metalloporphyrins as chemical mimics of cytochrome P450 systems, diversified analogues through automated oxidation chemistry, precision medicine, precision versus personalized medicine, a case study of Alzheimer's disease in precision medicine, and then finally translational chemical and systems biology drug discovery, before concluding. This article was written by Dr. Mukund Chorgard, Dr. Michael Liebman, Dr. Gerald Lushington, Dr. Stephen Naylor, and Dr. Ratnan Chugaturu. With so many contributors to this article, we'll be talking through their backgrounds at the end of part 2 in the next episode. If you've enjoyed this episode and want more from Drug Discovery World, then you can subscribe to the journal and get the latest issues delivered to you via post or electronically, completely free. Subscribing to the journal is the only way to get the full articles from the latest issues. Just head to ddw-online.com and you can subscribe there, as well as read further articles and view original PDFs of past articles, including this one, in our PDF archive. If you've enjoyed the podcast, then please do subscribe and leave us a review. You can also follow us on LinkedIn and Twitter for updates on latest articles and news. Thanks for listening, and we hope to see you for part 2 in our next episode.